Hello, world, and welcome to another episode of
Did You Know?
We love to talk about exciting subjects in science, medicine, robotics, astronomy, futuristic ideas, music, or anything interesting in the news. So let's get into it. Did you know in the news? Scientists grew stem cell mini brains, and then the brain sort of developed eyes. Now, this is an article that's put out by Michelle Starr on January 1st. It states that many brains grown in a lab from stem cells spontaneously develop rudimentary eye structures, scientists reported in a fascinating 2021 paper on a teeny human-derived brain organoids grown in dishes. Two bilaterally symmetrical optic cups were seen to grow, mirroring the development of eye structures in human embryos. This incredible result could help us to better understand the process of eye differentiation and development, as well as eye diseases. Neuroscientist J. Gopalakrishnan stated that our work highlights the remarkable ability of brain organoids to generate primitive sensory structures that are light sensitive to horrible cells types similar to those found in the body. These organoids can help to structure brain-eye interactions during embryo development to model congenital retinal disorders and generated patient-specific retinal cells types for personalized drug testing and transplantation therapies. Now, the article does explain that brain organoids are not true brains as you might think of them. They are small three-dimensional structures grown to induce pluripotent stems, harvest from adult humans, and reverse engineered into stem cells that have the potential to grow into many different types of tissue. Now, in this case, these stems are coated to grow into blobs of brain tissue without anything resembling thoughts, emotions, or consciousness. Such mini brains are used for research purposes where using actually living brains would be impossible, or at the very least, ethical, tricky test and drug responses, for example, or observing cell development under certain adverse conditions. This time, Gopalakrishnan and his colleagues were seeking to observe eye development. In previous research, other scientists had used embryonic stem cells to grow optic cups, the structures that develop into almost an entire glob of the eye during embryonic development, and other researchers had developed optic cups like structures from induced pluripotent stem cells, if I said it correctly. Rather than grow these structures directly, Gopalakrishnan team wanted to see if they could be grown as an integrated part of brain organoids. This would add benefit of seeing how the two types of tissues can grow together rather than just growing optic structures in isolation. So the article explains that eye development and complex processing and understanding it could allow underpinning and molecule bias of earlier retinal diseases. Thus, it is crucial to study optic vesicles that are primordium of the eye, whose proximate end is attached to the forebrain, essential for the proper eye formation. Previous work in development of organoids showed evidence of retina cells, but this does not develop optic structures, so the team changed their protocol. They didn't attempt to force the development of the purely-neural cells at the early stages of neurodifferentiation and added retinoid acetate of the culture medium as an eye aid to development. Their carefully tended baby brains formed optical cups as early as 30 days into development, with structures clearly visible at 50 days. This consistent with the timing of eye development in human embryo, which means these organoids could be useful for studying the intricacies of this process. There are other implications too. The optic cups contain different retinal cell types, which organize into neural networks that respond to light, and even contains lens and corneal tissue. Finally, the structure displayed retina connectivity to regions of the brain tissue. Gopalakrishnan stated that in the mammalian brain, the nerve fibers of retina ganglion cells reach out to connect with their brain targets, as aspect that has never before been shown in a vitro system, and is reproducible. Of the 314 brain organoids, the team grew 73% developed optic cups. The team hopes to develop strategies for keeping these structures viable on longer time scales for performing more in-depth research with huge potential. Optic vesicle containing brain organoids displaying high specialized neuronal cell types can be developed, paving the way to generate personalized organoids and retina pigment epithelia sheets for transplantation. And they finished off the article by stating that we believe that these are the next generation organoids, helping to model retinopathies that emerge from early neurodevelopmental disorders. Also, this research has been published in Cell Stem Cell. I will leave the link below for all the information. So what do you think about this, guys? Now, is this interesting news? Leave your comment. Let me know what you think. Until next time, stay safe and I'll talk to you soon.